Historically, developing a vaccine meant spending decades in a wet lab cultivating live viruses and running endless physical trials. But when a novel pathogen emerges, that empirical trial and error timeline is a liability. The response to that bottleneck is immunoinformatics. It is the specific interface where computer science intersects with experimental immunology. This intersection has created a methodology called reverse vaccinology. Instead of starting by isolating and growing a live physical pathogen, researchers begin by mining raw, high-throughput genomic data. By analyzing the pathogen's genetic sequence, scientists can predict its antigenicity and immunogenicity entirely in silico before running a single biological assay. Computational algorithms filter billions of potential molecular interactions. This reduction in the search space allows us to engineer highly complex, targeted vaccines with mathematical precision. To see exactly how these algorithms translate abstract data into a physical defense, we can look at the development of a multi-epitope subunit vaccine targeting the monkeypox virus. This is a diagram showing the complete computational workflow, plotting the exact route a candidate takes from a raw viral sequence to a fully simulated immune response. Following the perimeter, you can see how the pipeline forces the data through distinct phase shifts, progressing from a linear amino acid sequence into a three-dimensional structure, and finally into a kinetic simulation over time. This closed loop is designed to solve the central computational problem of biology, mapping the relationship between sequence, structure, and function. To understand how we can predict a human immune response using software, we must deconstruct this pipeline step by step, starting at the molecular bedrock. The process begins with antigen identification. A vaccine doesn't require the entire virus. Genomics data allows us to isolate specific, highly immunogenic proteins from the pathogen, like the L1R or B5R proteins in monkeypox. Here, the pipeline isolates these protein sequences. Predictive algorithms then scan the primary amino acid sequences to locate epitopes. These are the specific molecular sites that human immune receptors recognize and bind to. The software searches for two main targets: B-cell epitopes, which are recognized by antibodies, and T-cell epitopes, which drive cellular immunity. Predicting T-cell epitopes requires the algorithms to calculate exact binding affinities between the viral peptide and human major histocompatibility complex, or MHC, molecules. Only the sequences that score the highest for immunogenicity and test negative for inducing allergic reactions are selected to be combined into an artificial sequence. Those optimized epitopes are then assembled into a single artificial multi-epitope construct. To ensure these sequences don't fold into each other and block receptor access, computational linkers like EAAK are inserted. These maintain precise physical spacing between the epitopes. Finally, an adjuvant sequence is added to the artificial chain. This specific molecular tag is designed to agitate the innate immune system and force a stronger overall response. What we have now is a continuous, one-dimensional digital blueprint consisting of optimized immunological triggers. But linear sequence data has a strict biological limitation. Proteins only function based on their three-dimensional shape. If our artificial sequence misfolds, the vaccine is clinically useless. This requires structural immunoinformatics. We use homology modeling algorithms to predict exactly how that flat sequence of amino acids will fold into a tertiary structure. This ribbon diagram represents the predicted 3D physical model of our multi-epitope construct. To ensure this shape holds together, the structure undergoes molecular dynamic simulations. This calculates the physics of the molecule, testing how it behaves over time in a simulated physiological environment. These graphs output the molecular fluctuations. A stable trajectory confirms that the vaccine structure will remain intact under biological stress. Once stability is confirmed, the construct undergoes in silico molecular docking. This is the ultimate physical test to determine if the 3D vaccine will bind tightly to specific human immune receptors. This visual maps that structural interaction, proving the physical affinity between the vaccine construct and the target receptor interface. Confirming these structural physics in silico eliminates flawed designs immediately, saving the time and cost of synthesizing a candidate that would fail in a wet lab. Binding to a receptor is only the initial trigger. To fully validate the vaccine, researchers must predict the systemic biological cascade following that connection. They use agent-based mathematical modeling software, like CMSIM, to simulate the kinetic dynamics of the entire human immune system over a period of several weeks. This graph maps the simulated immune response. The black line charts the injection, a sharp antigen spike followed by rapid clearance. The true success metric lies in these rising curves, showing the delayed exponential generation of specific IgG and IgM antibodies following exposure. With systemic antibody generation mathematically validated, the optimized DNA sequence goes through in silico cloning. The digital code is inserted into a simulated plasmid vector, mapping out exactly how to physically manufacture the vaccine. This circular plasmid map is the final digital output of the process. By this stage, the candidate has survived atomic docking tests and systemic simulations. It is a validated, optimized blueprint ready for physical synthesis. Zooming out from this single monkeypox pipeline reveals an underlying question. What gives these predictive algorithms their accuracy? Machine learning models and molecular simulations rely entirely on massive, manually curated databases containing decades of empirical sequence and structural data. Every node in the vaccine pipeline pulls from specific repositories. Epitope predictions rely on the Immune Epitope database, or IEDB, while structural modeling draws parameters from the protein databank and the IMGT information system. Furthermore, these algorithms must account for human genetic diversity. Databases like IPD-MHC catalog variations in human alleles, ensuring a candidate vaccine will bind effectively across diverse global populations. This computational foundation extends well beyond designing infectious disease vaccines. This chart maps out the broader applications of the immunoinformatics field. The exact same predictive logic used to construct a viral epitope is currently being leveraged to design custom cancer immunotherapies, map the pathogenesis of autoimmune disorders, and execute precise HLA typing for organ transplantation. Moving immunology from the petri dish to the server converts the human immune response into a decipherable system of data. This computational approach drastically accelerates the path from raw genetic discovery to clinical therapeutics.